I would like to start with a 20-second audio. I mean, you will see what it is. So here.
And the mice will now not make their own antibodies anymore, but they will make nanobodies. And guess what they call them? Dixon, what do you think they call them, the mice?
Camel-like mice.
So, well, since this is Columbia, so I don't know whether how many of you guys have recognized the voice, but actually it's Dr. Dixon and Vincent, Bryony, and Alan talking about our work in the tweet this week in biology, and their title was "Can Nanobody Stop This Variant?" And well, if you want, you can go ahead and look at their discussion online. But today I'm going to talk about some more details and untold stories about this after this study. And so, like a lot of Aaron and Eli have mentioned, so a lot of us didn't work on SARS-CoV-2. Of course, nobody working on that before the pandemic, but we were able to jump into the study because there was some platform we already have. So it's the same case for us. Before the pandemic, we already have this so-called camelid mice or nanomouse and llamas, using them to study the influenza and HIV. But since last March, after the shutdown of NIH, so we have no option. We decided to jump into this SARS-CoV-2 study, so to continue the research. So the idea is simple. No need to talk about detail of this. SARS-CoV-2 virus uses spike protein to engage with the cell membrane, ACE2 on the same cell membrane through this RBD, the up and down conformation RBD. And the idea was to isolate neutralizing antibodies against this virus for protection purpose. And but since there's a problem with we are reasoning there's a problem with conventional antibody, since a lot of this virus, different virus, they have the heavy glycan shield over the fusion protein. And it's actually the case for also SARS-CoV-2, a lot of glycan on the surface of the amino spike protein. So then there's we can maybe an alternate way is we can try to use the smaller antibody that we in our lab, Dr. Cassella's lab in NIH is working on for a while, nanobody. So what is nanobody? Nanobody, normally it's from 20 years ago discovered from camels that camels have not only the conventional light and heavy chain paired antibody, but it has another type of antibody without the light chain that's called heavy chain only antibody. So what it happens is for the conventional antibody, there's VH genes, and then followed by recombination with Ds and Js, and also then to downstream constant Cs. After pairing with light chain, it gives a conventional antibody. And then sparse in this VH genes, there's some VHH, specific VHH genes for heavy chain only antibody. And this guys, after DJ recombination and to this specific C gamma 2 and gamma 3 that has no CH1 region, will produce this heavy chain only antibody. And then if you take out this VHH domain and DJ region, that is a small molecule called nanobody. And it's so small, it has a lot of advantage over the conventional antibody. And one of that is it's small enough to penetrate the glycan shield of the coronavirus, for example, in this case. So camel is one animal, and then there's also a bunch of camelid animals like apoca, geometry, llama, and guanaco and vicuña. They all have this type of conventional and heavy chain only antibody. So So the problem of making so-called heavy chain only antibody or nanobody with the llama or camelid animals is that those camelid animals are unsuitable for conventional laboratory facility. I mean, they are so big, so you have to raise them in the farm. So for example, this is our llama, Beijing. She's in Massachusetts, herbologic company in the farm. So, and also it's expensive to maintain those big animals. And for big animals, if you need to immunize them, you need a large amount of immunogen. For example, we always start with 1 milligram of protein and then boost with half milligram. That's a lot. That's enough for entire experiment with mice, if you want. And then after, a lot of times after project, companies would ask you to euthanize these animals, which is really like brutal. I mean, these are big animals, you wouldn't treat them maybe similar like mice. And then alternatively, you may, there's a way, maybe you can have a shark, using a nurse shark. Shark is another type of species that can produce heavy chain only antibody. But again, it's even less accessible to llamas, camelid animals. Or you can use synthetic libraries, as you have maybe seen in a lot of publications. But in our case, we are asking whether that was a lot quite a few years ago, we asked ourselves whether we can produce heavy chain only antibody or nanobody in mouse. From then we have a new species in our mind, nanomouse. So what is it? How do we make this nanomouse? So from Africa geometry and camel, we identified the 30 as different as possible VHH genes. And then we take these VHH genes, each of these VHH genes, upstream we put the mouse VH gene promoter, and downstream we put the recombination signal sequence. Yes. Then this would be a functional cassette, theoretically. And then we synthesize them through IDT and assemble them in a big mini gene and replace the entire 2.5 megabase VH gene of the mouse with this VHH mini gene cassette. And to make sure heavy chain and IgH heavy chain will not pair with light chain, we delete the CH1 domain from CMU and C gamma 1. And this way, we would expect IgM would be finally the IgM in this mouse would be heavy chain only, and it can switch to IgG1 also heavy chain only. So we called this a nanomouse. And after, a tremendous amount of work, we got this mouse, and here I'm showing you the just the profile of the IgM expression on the mouse. So for wild type mouse, you clearly see majority of IgM would be kappa positive, having light chain. And here is the heterozygous mouse, nanomouse. You can see this IgM. Some of them are kappa positive, and you start to see a lot of a big population of IgM without the light chain, without Ig kappa. We also checked lambda. There is no lambda chain expression either. And then because we have 30 different VHH genes. This is from homozygous mouse. We sequenced the usage of the deep sequence and checked the usage of the VH genes. And all of these 30 are being used, some at very low level, some at the relatively higher level. Overall, it's we have a lot of VH being really well used. Then how about the immunization response? So we immunize nanomouse. You can see a very nice germinal center reaction, germinal center population, and which is also, you can see IgG1 switching in the germinal center cells, and which is really like comparable, if not more than the wild type control. And like I said, we studied before pandemic, we worked on already some other immunogens, and like here, HIV aminoprotein. And there we immunized mice, and before and after mice immunization, we noticed a lot of somatic amplification in the VH genes. Overall, this is telling us with this mouse, although without light chain, and they behave really well. I mean, B cells. So then we started this SARS-CoV-2 study, by immunizing Beijing and nanomouse with SARS-CoV-2 RBD protein, followed by a spike boost. This is to make sure skew the antibody response to the native conformation of RBD. And then it's very simple now. You can make a phage library. You don't need to do like a sorting or whatever. You can do a phage library, and that gives you all possible repertoire after immunization. And you do phage screening, and then neutralization test for those candidates. So out of a lot of candidates, we eventually identified or focused on 6 antibodies, 4 from Llama, 2 from nanomouse. And we made this nanobody 15 kilodalton, and then we also fused human IgG1 Fc domain to make it like bivalent, and also to even theoretically boost the activity of this nanobody. We put them in even trimer form. And you can see here is the example of MB12 from monomer, bivalent, and the even trimer version, the neutralization potency against SARS-CoV-2 pseudovirus increase. And then this is IC50 of this specific MB12. And the best one we have is an MB19, 9 picomolar even. IC50. And why there is the improvement of the IC50 over the trimerization? So we did a bilayer BLI assay. So it works this way. You load a RBD protein on a biosensor and check the antibody, your nanobody binding and association and dissociation rate. So this is what it works. So you in this phase, you load nanobody, and then you put the probe into a solution without nanobody, and you start to see the dissociation, and you calculate the dissociation rate. And then after trimerization, you can clearly see the dissociation is really, really slow. Basically, after the trimerization, nanobody do not come off from this targeted protein, and we have very, very good KD value. And then comes the, it was in early January this year, we have this variant of concern, and like but now we name it alpha, beta, gamma, and they have because a lot of us were focusing on the RBD region, so they share actually mutations in RBD region, and also the more recently, the most concerned one, Delta variant, it also has this shared region in the mutation in RBD region. So we asked whether our nanobody can neutralize those variants. First, of course, starting from the pseudovirus. And of course, monomer and the trimers, they neutralize wild type really well. And for this specific mutations in RBD region, especially this KEN mutation, where you combine these 3-point mutations. This KEN basically is in South Africa variant. So especially in this KEN, mutation kills the activity of the monomer of all 4 Llama-derived nanobody. But the nanomouse-derived AB12 and 30, although the neutralization activity is low, but it's not affected by the mutations. So I get back to this later. And then very, very impressive is when you put this in trimer form, and although it's blocked by mutation, now the trimer can overcome this escape. it's very interesting to us. And so then, of course, we want to ask whether they can neutralize a real virus. And we tested this with David Ho's group. Of course, this South Africa, that time, I mean, it was early March. That time, Delta variant wasn't as important as now. So South Africa variant got nicely neutralized. And recently, Dr. David Ho's lab people helped us and tested the Delta variant. Again, it's our nanobody can nicely neutralize the Delta variant really well. Of course, it's a trimer version, and of course there's a UK variant and Brazil nicely neutralized. And the neutralization IC50 still stays at picomolar or nanogram per ml level, really potent. Still rivals the most existing human-derived antibodies. And this here, I have to, the untold stories, we have to have special credit to Nature reviewers who insist we have to include this authentic virus neutralization data. And then here, because Llama-derived nanobodies are blocked by RBD mutation, and nanomouse are not. So derived nanobodies are not. So they probably recognize different epitopes on RBD. So we decide to do two experiments to see the mechanism of that. One is still the BLI assay. So what we do is we load, for example, one antibody to RBD, and then we ask whether other nanobody can load upon the existing of the, for example, MB15. What we see here, when you load Llama, for example, blue colored nanobody, none of other Llama-derived nanobody can load to this sensor anymore. But the nanomouse-derived ones can, and it's the same when you do the opposite way. When you load nanomouse-derived nanobody, and for example, you load 12, 30 will not come anymore. But then, Llama-derived nanobody will still bind. So definitely they are binding to different epitopes on RBD region. And then we tested we checked the structure of this binding complex and try to find the mechanism of this. So here is the MB12 and the 30 of the nanobody in complex of RBD, in complex of spike trimer. And what you can see here is MB12 and 30, those two are derived from nanomice. They both bind to a region away from this ACE2 RBD interface, binding interface, and where those mutations are, important mutations are happening. So that explains why their neutralization activities are not affected by this variant mutation. And the binding of the nanobody to RBD both clashes with the ACU binding to RBD, and that's possibly the mechanism why they neutralize. And I will come back to this later. There's some other explanation for this neutralization mechanism other than ACE2 blocking. And then for Llama-derived nanobodies, all of these 4 nanobodies, they bind to actually the ACE2 RBD binding interface, making the like RBD all down, all three RBD down conformation, or in this 3 case, 1 RBD up and 2 RBD down conformation. And I have shown you this MB12 and MB30, they bind to the domain away from the ACE2 binding interface. And this region, how about the conservation of this? We tested we analyzed the 9 different virus spike protein, the conservation of the amino acid, and to figure out that this region is really not conserved, and the region bound by MB12 and 30 is really conserved among this virus or sabica virus. And again, so we tested, we decided to test whether our nanobody can neutralize but those two nanomyce-derived nanobody can neutralize, for example, SARS-CoV-1 or bat coronavirus, WB16, and as predicted from the binding, they neutralize really well. And nanomouse, llama-derived MB56, which bind to the less conserved domain, does not neutralize those two virus in the pseudovirus condition. And this was done in HIV-based pseudovirus. And then nicely, David Ho's group, they tested another pseudovirus system in VACV-based. And we tested the SARS-CoV-2 itself and the SARS-CoV-2-like virus, and both from bat and pangolin. Again, our nanomouse-derived one could conserve the binding to conserved domain, they neutralize really well. And for SARS-CoV-1 and SARS-CoV-1-like virus, is similar. We saw similar results, confirming the mechanism of derived from the structure analysis. So here I'm showing you the based on 50 at that time 53 available human neutralizing antibody structure, but we plot where do they bind, where do those human neutralizing antibody bind. Most of them, they recognize the less conserved ACE2 RBD interface, and only maybe one or two of them bind to recognize the conserved domain. And here I'm showing you from Llama the derived nanobody when we make it a trimer version. It can, of course, again, recognize the same epitope, but it now overcome the mutation of the variants and overcome this escape by enhanced ability possibly. And then on the other hand, from the nanomouse, nanobody from nanomouse will overcome this RBD mutation by recognizing a conserved domain. And 1 month after our publication, there were 2 more papers, or even a lot more, I believe. This is the 2 I'm showing you here, talking about the human antibodies that isolated, neutralized antibodies isolated from humans, and that can achieve broad neutralization across the septic virus. And very recently, there is 1 more that human antibody that recognizes the stem helix region that can even extend the neutralization activity to the beta coronavirus.
Jianliang, sorry to interrupt. This is a 2-minute warning.
Yeah. Thanks. And then for the RBD conserved domain neutralizing antibody, there's 1 paper recently published claiming or saying actually they are neutralizing through interacting with lectin, which enhances the SARS-CoV-2 infection. And I think this is the last slide. So I have some additional information. So nanomouse, since our publication, a lot of people request the mouse from us. It's a nice tool, we believe. And so we deposited this to Jackson Laboratory, and they are working on this, and then should be available very soon. And anybody interested in this can inquire to the Jackson Lab. And one more thing is, I'm available right now. So this was me last June, isolating, working on the Llama blood in the pandemic. So I'm now seek to establish the independent lab to develop antibody therapeutics again, re-emerging virus. So with that, I would like to thank a lot of collaborators and Dr. Capella's lab and Dr. Peter Kwan's lab people, and Michelle's and Theodora's lab and David Ho's group, and also our NIAMS co-facilities and or a lot of collaborators. And thank you very much.
Thank you very much for the great talk. If I could lead off with a very simple question. Can you comment on the cost and the time of to production for a nanobody if you have a llama on one hand or a nanomouse on the other?
Yeah, so one important thing is, for example, this SARS-CoV-2 spike protein, it was at the early time, it was very difficult to produce in large amount. So in my case, I already immunized nanomice, I mean, a month or two, but I couldn't still have enough protein for llama immunization. So it was if you have enough immunogen, that's not a problem. It's easy for timing. And for expense, I think nanomouse with mouse is definitely cheaper and more convenient, definitely.
And when you screen the phage library, how many potential candidates are involved in the initial screen that led down to the two that you studied further?
In nanomouse case, well, you randomly after screening you randomly pick. So in this case, I remember it's not many. A lot of them are showing up repetitively. I mean, not 20, 30. I mean the unique sequences, but you sequence a lot. You sequence maybe 100 or 200 of this. But that's after several steps of phage screening.